And so we actually received one of the very first Dean's Awards, the Alliance Awards, and that allowed us to build the Kentucky Research Alliance for Lung Disease. And that gave us money to start a pulmonary critical care biobank. So usually what happens is when someone is being evaluated for transplant, we start to follow them. We start to get permission from the attendings. We talk to the patient once they're going for lung transplant. And then if they consent, which everyone consents for the biobank. I've yet to have somebody say, no, you can't have my discarded tissue. And then once the transplant actually occurs, then I go to the operating room or surgical pathology and get the lungs for the lab. And because we had already had this in place before COVID, we actually were able to compare what happens at the molecular level to a lung that is scarred or fibrotic post-COVID to a lung that is scarred or fibrotic due to other lung diseases. Other universities who don't have pulmonary critical care biobanks like ours aren't really able to do this type of research. And the advantage of being, at UK and being able to have great partnerships with the clinicians and to be translational is when experts in the field contact you and say, hey, I have this question, being able to have these samples have really been able to advance the science. Dr. Lisa Cassis, the VPR, she's an expert in ACE2. SARS-CoV-2 is the virus that causes the disease COVID-19. It uses the ACE2, the protein that Dr. Cassis studies, receptor. And so she was working with Dr. Chris Waters. He's a basic scientist who studies pulmonary fibrosis and lung disease and acute respiratory distress. So they were talking, and Dr. Waters said, hey, we have this biobank through our alliance. Let's ask Jamie if we have samples. And so I said, yeah, we actually have done COVID lung transplants. And so because Mr. Hoover had donated his lungs, I was actually able to match other donors in the biobank, age, sex, race, ethnicity matched donors who had transplants, but were not COVID. Like these were due to other fibrotic diseases. And you can see at the molecular level, there is a difference in the ACE2 function in the patient who had COVID versus the one who had fibrosis due to a non-infectious cause. And so Dr. Cassis, Dr. Waters, and I and our team have started to look at this with the other COVID transplant samples we've gotten, and hopefully being able to now take what we see clinically by following these patients during active COVID, post-COVID, and into the transplant space to having the clinical samples and data, and then coupling that with the mouse models and expertise of Dr. Waters and Dr. Cassis, we may be able to figure out some new pathways that would maybe prevent fibrotic changes due to the virus.